Welcome back. This is video 2. I hope you're thinking a little bit about inheritance patterns for organelles. And now we're going to look at where they actually came from. I want to talk about endosymbiotic theory, the theory of endosymbiosis. So I don't usually do this, but we're still in the first part of the textbook here. So this is found in 9.2. If you're reading along in the textbook, it actually has some handy dandy like patterns and questions to follow. I think that I have summarized it pretty well here, but if you need the textbook, absolutely, or ask me questions. I also think you covered this in Bio 1. All eukaryotic organisms stem from an ancestor that has at least one event of endosymbiosis. Endosymbiosis is that ancient event, and maybe even more modern, where a cell that could engulf and create a vesicle or bring in something that it was eating, engulfed in a bacteria or other things we'll look at. We know that the first event that we define eukarya at is the event where we now know it was an archaea that engulfed an aerobic bacteria and led to a mitochondria. The thing about endosymbiosis is that it occurs multiple times in the tree of life. It often occurs in series. I want to start out with this idea for endosymbiosis that mitochondria and chloroplast result from endosymbiosis of a cell engulfing a bacteria and that bacteria moving inside of the cell and kind of still doing what it's doing. Over many, many years, and billions in this case, over billions of years, what has happened is those relationships have gone from being a symbiosis to an obligate relationship, where it's obligated symbiosis. A symbiosis is when two different organisms live together very intimately. Obligate means they are required. And what has led to that obligate relationship is that some of the genes that the mitochondria needs to live are being some of the proteins that the mitochondria needs to live are found on the nuclear genes of the cell that hosts the mitochondria. Now they can't live apart from each other. They're obligate. Mitochondria are really the first result of this endosymbiotic event, at least the one that was successful. We know that the thing that was engulfed was an aerobic, meaning it can do cell respiration. And we know that because that's what mitochondria do for us, most of cell respiration. And it was an alpha proteobacterium. Proteobacterium is a group of bacteria, like a kingdom of bacteria. The proteobacterium include a lot of significant bacteria for doing things like converting gases into organic materials. The aerobic alpha bacterium include things that can do things like nitrogen fixing, the nitrodomus bacteria, and other bacteria that are really significant in our ability to just live on planet Earth. They make life for us livable. In the case of the alpha proteobacterium, the one that was engulfed in and became a mitochondria, leads to the ability of eukaryotes being able to do things like multicellularity and complex systems. All eukaryotes come from this original lineage of having a mitochondria. Some eukaryotes have subsequently lost their mitochondria, but their ancestors all had them. And so this is from an older textbook, but it still checks out. This proto-eukaryote we know now was an archaea. Or an archaeobacteria, depending on what you learned in bio. They had maybe had a nucleus and an endoplasmic reticulum. We know they had the ability to engulf things and create a vesicle. This thing ate an alpha proteobacterium that was aerobic, and it moved in, and at some point in their history, they went from being just sharing a space to actually obligate with each other. This thing is still heterotrophic, meaning it still eats for a living. You still are too. Your ancestors were heterotrophs, their ancestors were heterotrophs, all the way down before you were even a eukaryote. That's how the mitochondria got inside. And our lineage actually as animals and fungus and slime molds and a bunch of other things actually continue on this pathway. There's another possibility here. This organism, some of our not ancestors, but I don't know, aunts and uncle sisters, went from being a heterotrophic eukaryote with mitochondria, and while they were eating, some of them ate a photosynthetic bacterium. This has led to the development of chloroplasts, plants, algae, and we'll look at some other things as well briefly, are the result of two endosymbiotic events where bacteria were engulfed. Chloroplast, I have the picture here, so a little bit, chloroplast resulted from the eukaryote eating another bacteria. That leads to modern plants and algae. That thing here, so here it is, modern plants and algae, is minding its own vision. Let's say this is an algae. It could be a red algae or a green algae. These two are both related to plants. They're both little photosynthetic fellows and they're doing their own job. This thing is minding its own business. And a eukaryote with just a mitochondria comes along and noms it. Well, now there's an algae inside of this algae. Inside of this one has its own mitochondria and its own nucleus and its own endoplasmic reticulum, but now it also has inside of a vesicle, it has another eukaryote, and that eukaryote has a chloroplast and its own mitochondria. This is actually what's called secondary endosymbiosis. More like inception. There's an algae inside of a eukaryote. So a eukaryote inside of a eukaryote. So a eukaryote, prokaryote. So the prokaryote is 2 levels down. This example over here, where the eukaryote is eating a bacteria, resulting in a eukaryote that can photosynthesize because it has a legitimate chloroplast. This is primary endosymbiosis. And we can write primary as that, or this, or I never write it like this. Primary, mostly because it looks like an L. Primary endosymbiosis and secondary endosymbiosis can occur in series. We call it serial endosymbiosis. This is series like a TV series or a serial killer. One event followed by some waiting time, followed by another waiting time. Primary endosymbiosis is when a bacteria is engulfed. So the cyanobacterium, the photosynthetic bacterium that led to a chloroplast, or the alpha proteobacterium that led to a mitochondria, both of those are instances of primary endosymbiosis. Secondary endosymbiosis is when a eukaryote is involved. So for this organism, it's a eukaryote, which means back here, let me draw it. There's a moment where its ancestor engulfed an alpha proteobacteria before it became a eukaryote. This is primary endosymbiosis, followed by primary endosymbiosis. It's not the order, it's who's being eaten. These two primary events in this storyline were followed in series by an event of secondary endosymbiosis, where a eukaryote was engulfed by another eukaryote, and now the bacteria are 2 layers in. This is primary to secondary, and there's a lot of things out there in the ocean, particularly that result from this. We say that the thing inside of it then, and it's almost always photosynthetic, this organelle is a plastid coming from chloroplast. A lot of them serve many functions beyond photosynthesis, but outside of the scope of this class, because we're not going to worry about their genetics at all, but kind of an interesting thing that you can keep adding on cells, kind of like we added on plasmids inside of the bacteria to add additional functionality. The other thing I want to point out here is the serial endosymbiotic theory suggests that mitochondria and their ability to do cell respiration evolved once. The fact that most living things can do cell respiration is a result of either being an aerobic bacteria that is a direct ancestor, vertical transfer, from that original evolver of that complex system, or in most cases, the result of endosymbiosis, where a cell engulfed and incorporated another cell to add on functionality. And that makes a lot more sense if you look at the span of evolution that's involved here. Evolving something as complex as cell respiration is unlikely to happen twice in exactly the same way. There's just too many variables. But if everybody's taking that one individual and then using them in their own way, that means that something like respiration or photosynthesis later can rapidly spread throughout all the species that are involved without having to wait for random events in evolution to take place. All eukaryotes have endosymbiotic relationships, and inside these endosymbiotic organelles, mitochondria and chloroplast, I can see that they have their own DNA. This is a xenopus frog. I love them. We actually know a lot about them, and also, who could forget that face? Xenopus frogs have been really well studied for their mitochondrial DNA. When I refer to mitochondrial DNA, I put an MT in front of it to indicate that it's in the mitochondria, and this is what it looks like. It looks very much like bacterial DNA. This is lettuce. Lettuce and its chloroplast DNA have also been really well studied. You'll notice we put a CP in front of the DNA to indicate that it's chloroplast DNA, not nuclear DNA. Chloroplast DNA, the CP DNA, is quite large compared to mitochondrial DNA. This is 100 to 225 kilobase pairs. A base pair is when I talk about an A and a T bonded together, or a C and a G bonded together. So it's one base pair. They have 225,000 base pairs in a chloroplast DNA possible. And we actually see multiple copies of that DNA molecule per chloroplast. They include a lot of the code for doing actual photosynthesis. Plants, the eukaryotic part of plants, can't photosynthesize at all. They entirely rely on their chloroplast to do that. The code for rubisco, which is the protein it's the enzyme that can grab carbon dioxide out of the air and fix it into the Calvin cycle, is included in this chloroplast molecule. I would argue as an ecologist that is probably the most important protein on planet Earth, because all the rest of us can't photosynthesize. And so there's no way for us to get carbon dioxide, a non-organic gas, and sunlight together to incorporate that into a form that living things can use. This enzyme is really what makes complex life on our planet possible, and it's found in the chloroplast DNA. When I look inside the chloroplast, I find that they have something that looks very much like bacterial DNA and 70S ribosomes. You might remember from micro or other classes that ribosomes are pretty, everybody's got one, all living things, because that's how we read our DNA and turn it into a protein. But our DNA as eukaryotes are 70S. This is the number of protein structures that actually make them, incorporate it with RNA. We have that in a couple lectures. This looks more like a bacterial ribosome. Let me show you a little bit about what I mean here. Mitochondria are making some of their own genes, and the way they make them is polycistronic. many on the same side of the gene. This is really common in bacteria. It's not really found in eukaryotes as far as I know. We mentioned it a little bit the other day when we started the P-glow discussion when I talked about operons. In bacteria, often there is, we talked about the fact that there's a promoter and an operator. The operator controls for how the RNA transcription mechanism, so the proteins involved in transcription, they are allowed to access gene 1, gene 2, and gene 3. If the operator is free, RNA polymerase will bind to the promoter and transcribe all 3 of these genes at the same time. This is polycistronic. And in bacteria, it means that they encode a transcript that covers multiple polypeptide sets of instructions, and that's clipped apart later as it goes through the process of translation and protein folding. In eukaryotes, we don't have these operons or these genes that are all coded together. We code for one gene at a time. We'll highlight a little bit of differences in that later. Mitochondria have polycistronic genes, which suggests that they work the way that bacteria does, which suggests they're related to bacteria. They have bacterial type ribosomes, kind of like the chloroplast. They use bacteria type enzymes to do a lot of the things that they do inside of their little mitochondrial cell. They also use a lot of eukaryotically made proteins to maintain themselves. We, both us and bacteria, or us and our mitochondria, use enzymes to copy and transcribe and translate the codes for our DNA and turn them into proteins. But we use different genes and slightly different versions of those proteins to do it. This fellow is using the bacterial versions. We're using a completely different eukaryotic version. And so it's other additional support that these are basically coming into our cells at some point in the history of the evolution of life on Earth as bacteria being incorporated horizontally. Our mitochondrial DNA is in our mitochondria. It belongs to the mitochondria. It has about 16,000, so this is like 17 kilobases of DNA information and codes for 13 proteins. All of these are involved in aerobic cellular respiration. They allow you to respirate. When you interrupt that, or if those genes are bad, that's bad. Respiration is what provides us with accessible energy. It converts sugar into ATP, which is the actual cell energy that our cells use in order to function and maintain themselves. Mitochondrial DNA, because it comes from an ancestral history that is bacterial, does not have any histones to protect structures. I have not yet talked about this, but we will have it in the next couple weeks. Our DNA as eukaryotes is, and I really only have one color at a time here, our DNA is twisted and wrapped and bound together with these little histone proteins that maintain the structure but also keep it from unraveling and tearing. Prokaryotes don't have that. Mitochondrial DNA doesn't have that. It means it's more fragile and likely to suffer breakage. And there are a lot of basically things that can damage it. If you're a mitochondria, your job is to draw in oxygen, and in the presence of oxygen, convert sugar into, pyruvate into a usable form for the cell, which means you're constantly being bathed in oxygen and other reactive oxygen species. Those things are bad for you. Oxygen's bad for you. It tears up your cells, it causes mutations, it causes damage to the cell. Oxygen's very electronegative. It pulls things apart. It's highly reactive, and which means that the mitochondrial DNA is sort of under assault from all sides from many things that are damaging it, without any of the defenses that eukaryotic DNA has. It means that those codes can generate more mistakes than you would expect. And they do reproduce. They don't live eternally inside of yourself. So the mitochondria that you have are different than the ones you were born with, but they are descendants of the ones you are born with. When you were a fertilized egg, you got all of your cytoplasm and organelles from your mom. The function of the egg is to provide all of the nutrients and the enzymes and the structures that you need to develop a growing organism. The sperm provides the other half of the nuclear material so that the organism ends up being diploid, which means that the mitochondria that you inherit or that you will pass on if you are making eggs varies inside of your egg, because they're not what you started out with exactly. They're descendants of your original mitochondria. There's mutations and problems that arise on the way. All of those mitochondria are not clones of each other. They're variations on a mitochondrial theme. There is heteroplasmy, which is supposed to be red, because it was red in the beginning of the story. Heteroplasmy, meaning what you pass on or what you inherited from your mom, may be variations in mitochondria. Adult cells generally have a combination of functional and non-functional mitochondria, and as long as you have enough, it's fine. It's fine. This is dosage we talked about before. You just need a certain amount of cell respiration to function. Your book goes into, at this point, and absolutely this can be, if you're still thinking about what you want to do for your project, this can be your project. Your book goes into a variety of different mitochondrial diseases. I picked myoclonic epilepsy and ragged red fiber disease to show you very briefly, but not to step on someone's toes in case this is what you want to do for your presentation. It has a pattern of inheritance that is absolutely tied to mitochondrial maternal-only inheritance, and it causes the loss of muscular coordination, and these here are very characteristic red, raggy-looking muscle fibers. This is where it actually gets its name. We are now starting to understand the role of mitochondria as people age. As you age, it's more and more likely that you make mistakes in your nuclear DNA, but also in your mitochondrial DNA. And dysfunction in mitochondria have been tied to everything from anemia, which is sort of the inability to ultimately deliver oxygen to your blood cells, blindness, type 2 diabetes, Alzheimer's and Parkinson's disease, autism. We're trying to tie it to a lot of things. I think there's been a lot of very good recent research tying it to Parkinson's disease. There are a variety of treatments that we can give. Your mitochondria are not you. And so it has been possible in the past to give people new mitochondria, but in a cell, right? I don't have the ability to zoop you through a machine and replace all of your mitochondria, especially at an age where you're not making a lot of new cells. As a zygote, sure. As an adult, that's a billion cells that I have to worry about replacing the mitochondria for. About 1 in 5,000 humans have a mitochondrial-based disease or have a risk of mitochondrial-based disease. We actually can detect this with genetic testing. We can look at the genes of the mitochondria. We know that we can replace the mitochondria in a person or a monkey that have bad mitochondria with good mitochondrial by using a maternal mitochondrial donor. What that means is that we take the egg of the mom who wants a baby, but she has bad mitochondria, and we remove all of her bad, sick mitochondria in the egg and replace it with mitochondria from a healthy donor. That egg is then fertilized. That egg has mom 1 nuclear DNA, mom 2 mitochondrial DNA, and dad's contribution from the sperm, parent 3, we fertilize the egg, and that egg can develop normally and will have mitochondria from the donor mom. technically that means genetically that offspring has 3 parents. One of them is just donating mitochondrial DNA. It's called mitochondrial replacement therapy or 3-parent therapy. The book talks about it in rhesus monkeys. This has absolutely been done in humans. This has been done successfully in humans, and if we know that mitochondrial mutations are a problem, this is a completely accessible technique as long as it's done to the egg. This is, again, not reasonable to do with somebody and they have millions of billions of cells in their bodies. We don't have that technology. I can't replace all of your mitochondria. And one more example, just to talk about the fact that how could anyone have ever figured this out? I want to talk about the maternal effect and how it kind of muddies up our understanding of gene expression in general. Sometimes the mother's genes determine what happens to the babies, regardless of the baby's genotype. In this case of maternal effect, I want to talk about these snails. These snails spin either right or left. The difference between right or left here is here, the way the snail shell actually spins during development. It has to do with the way that the cells align during very early embryonic development. It results in them having opposite spins of their shells. The effect is that as the babies develop, they are primarily affected by proteins in the egg that their mom gave them, not by their genotype. So their spin is determined by proteins their mom made, not by proteins that they made, which is one step away from what all of the other expression we have talked about. I want to show you a little bit, don't get too caught up in this, but look for the pattern. This is a right-handed and a left-handed snail. When I cross them, they are hermaphrodites. And that means that each one of them is giving egg and each one of them is giving sperm, and I'm getting a lot of babies. So if they got egg from mom and that egg was dominant, it was telling it to spin right, and their sperm from the left-handed one, that's their dad, they have a right-handed spin. Okay, that's not shocking, right? Because we can assume that right D is dominant here. These babies are also heterozygous, but they got their dominant from this snail, but it was sperm, because in this case, this snail is acting as dad. They got sperm from dad, and egg gave lowercase, the recessive allele. They look like mom. They are heterozygous. They have a dominant right spin allele, but because mom was left-handed, they are left-handed. Because mom was right-handed, they are right-handed. This is a right-handed and a left-handed snail. When they go through self-fertilization, mom's right-handedness is a gene that she is making in her egg, right? This snail is making, in her eggs, she's making some dominant and some recessive. She's making heterozygous genes. Because she has dominant alleles, she is making a lot of right-handed genes in her eggs as she produces. And so her babies are all right-handed spin. Her babies are all right-handed, but she's passing on her dominant or recessive allele. So sort of without drawing out the whole square, because you know it by now. She has a homozygous dominant. I'll do it anyway. Heterozygous and homozygous recessive that she can make. She's self-fertilizing herself, so she can make babies that are diploid. These babies have an expected 1:2:1 ratio in genotypes, but they don't have the 3:1 expected phenotypic ratio. They're all dex. They're all right-handed spin. These babies make their own babies. So we self-fertilize this one together. This one only can pass on homozygous dominant. Its baby's homozygous dominant. Sure, that's exactly what we expect. This one can pass on heterozygosity. Its baby's also right-handed. This is not shocking. This one is left-handed looking, it carries no genes for right-handedness. When it makes alleles, when it makes proteins, it only makes left-handed proteins into the eggs. Its baby looks left-handed. In this case, the babies are influenced by the genotype of the mom, not by their own genotype. So we do ultimately see the 3-to-1 ratio that we expect in phenotypes, but it's skipping a generation because we're seeing the expression of this one generation later. And we can talk about this more. I have some examples for you to look at in the homework and beyond. Consider that this is just delaying the expression of the phenotype one generation behind. And that's what helped me come to terms with how strange this pattern is. I've got a couple practice with me questions for you. In Chlamydomonas, there is a one-parent inheritance of mitochondria. It is the MT mating type negative parent that passes on the mitochondrial DNA. And for their chloroplast, the MT positive parent, the one mating type positive parent, passes on the chromosomes from the chloroplast. I have an MT negative, a mating type negative mutant that's discovered that's resistant to an antibiotic. When crossed, the offspring are sensitive. So it's here, it's resistant, but all of the offspring are susceptible. Is this trait inherited from the mitochondria, from the nuclear genome, or from the resistance trait, or from the chloroplast? I'll give you a second. Sorry about my bird today. I don't know what she's got. She's just so spunky today. I'm trying to keep her in control, but she's running all over the keyboard and screaming. She's happy. Okay, I'm going ahead. This is coming from the chloroplast. The mating type parent that is positive passes on the chloroplast DNA. The negative one passes on its DNA in the mitochondria. If it was mitochondrial, the resistance, I would see, because it's tied to the mating type negative parent, if it was passed on through the mitochondrial DNA, I would expect all of the babies to be resistant. Since none of the babies are resistant, I expect that it's from the mating type positive and it's chloroplasts. If it was nuclear, I might see 50-50. Maybe it's passing on its resistance or not its resistance. I might see 100% passing resistance on. This pattern in this example is only going to be seen in a chloroplast inherited pattern. Practice with me too. This woman has a rare heritable, means she can inherit and she can pass it on, defect in the bioenergetic function of her mitochondria, basically the living energy functions of her mitochondria. Her dad did too. If her husband does not have the trait, what can you predict from this? And I'll give you a minute. And you can pause me, but I'll give you a hint here. Don't assume just because the question is in the end of chapter 9 that it necessarily has to do with mitochondrial or chloroplast or maternally inherited. I now it's free for me to use all the inheritance patterns we have discussed in all the questions. Oh, Alice revealed the clue. Alice is the bird. Her trait is following normal Mendelian patterns. She has inherited from her father. So it's not mitochondria. It's not chloroplasts because we don't have those. And we're assuming that this is a human woman, not a plant woman. Her husband doesn't have the trait. Yeah, it's Mendelian patterns here. This is a Mendelian question. So here's a snail. This snail is a right-handed coiled snail. It's the result of a cross between two snails. So it's two snails that are both making sperm and both making eggs. Snail is genotypically left-handed. What is the genotype of the snail that provided the egg? Don't just say you notice in some of them on the homework it says you can't determine from the information you have. In this case, you can say something about it. She's right-handed, but she inherited little d. Little d. Okay, and pause me if you are still working on it, because I'm going to go on. Don't step on the keyboard. Her mom, the egg provider, must have been heterozygous because she's expressing right-handed, and she expresses her mother's genotype, which is right. But she inherited two little d's. She must have inherited a recessive D from dad, but she also inherited a recessive D from mom, which means mom looked right but was hiding left. And no, on the exam, I'm not going to tell you what the inheritance pattern is. I might lead you to it, though. This is no way this is a Mendelian pattern, because here, her genotype is different than her phenotype. Now, in addition to making a key and following through that pattern of figuring out what do the different notations mean, I need you to highlight for each thing as we go through what is the phenotype, what is the genotype, and do they or do they not match? If they do not match, that is a clue that you are looking at some sort of extranuclear inheritance. With that's the last one. Yes, that's the last one I have for you today. We'll keep it, and I'll just boot back to it. Take a look at these. We'll talk about it in lecture. If you come on Tuesday, right now, we're planning on doing more PGLOW stuff, and then we will work on our extra questions while we set up stuff for PGLOW. And if we don't have time, absolutely email me, ask me questions at any time, and make sure that you understand. Okay, I'll see you soon. I'll talk to you later. Bye-bye.